Okay. Hello, everybody. Back for the second debate, which is about the microbiome is a therapeutic target in MS. I'll give you just the brief capsule, which you can also read in the program online. So environmental factors are thought to trigger and perpetuate inflammation in people with MS. And some preclinical data, as well as clinical actually, have shown that gastrointestinal and microbiome environment may play a critical role in regulating immune response, in autoimmunity in the CNS. Fecal transfer has been successfully used in inflammatory bowel disease, and in terms of safety also in neuroinflammatory disease. And it's not a stretch of imagination to think that similar interventions could become available for people with MS. So do the preclinical and now also increasingly clinical data provide sufficient plausibility to pursue this route of inquiry further? And what is the magnitude of the effect of the microbiome in MS? So what role do approved DMTs play in microbiome biology? All these questions are related and will be debated after the first vote. And I'm going to read out the question again, which is, or the motion that we're debating. The microbiome is a therapeutic target in MS. After the vote, I will introduce your speakers before they give their presentations. So fire ahead with the vote, please. Right, so where are we here with the vote? 58 in favor, 42 against. And so this is our starting point. And we have really two formidable speakers here, both friends as well. First one is Professor Chris Constantinescu. He is an emeritus professor of neurology from University of Nottingham, and now an attending neurologist at the Cooper Neurological Institute in Camden, New Jersey. He has, in addition to his clinical activities focused on MS and other neuroinflammatory diseases, he's been an investigator in many MS trials, phases 1 through 4, and he has an interest in immunology, imaging, and epidemiology, and is also teaching now at the Cooper Institute. He did his MD at Boston University and a PhD at University of Pennsylvania. He has been on the faculty for quite some time, in Nottingham since 2000 and Cooper Medical School since 2022. His opponent is Patrick Vermersch, who graduated in neurology in Lille University in France, where he's now also the head of one of the departments of neurology with a focus on MS and other neuroinflammatory diseases. It's mainly concerned really with neuroimmunology and markers of disease evolution. Patrick has pursued training in basic research, including cellular biology, obtaining a PhD in the field of Alzheimer's and other neurodegenerative diseases before turning to MS. He is the Vice President of Research in the fields of life and health sciences at the University of Lille. and he set up the first network of MS centers in France from 2000. And he is the PI of numerous clinical studies and CTIMPs, and is very well published. So really a wonderful debate ahead, I expect. And I suggest we listen to both speakers now in succession before the discussion.
Ladies and gentlemen, thank you for the opportunity to be here at CONI, albeit virtually again. I'm very pleased and honored to debate against a very reputable neurology professor, Professor Vermeersch. But I do hope to convince you, or to actually show you, because I'm sure you're already convinced, that the microbiota is a very important therapeutic target, already is a therapeutic target in multiple sclerosis. These are my disclosures. Thinking about this topic, I came up with a number of questions, part of which have been addressed here at CONI several times, and for which microbiota can provide an answer. So I remember the first CONI 16 years ago, I think, in Berlin, when the hot topic was, is MS an immune-mediated disease, or is it primary neurodegenerative disease? And at the time, that was a debate. It was a very valid debate, very hotly debated. At the moment, it's obsolete. We all know that the immune system should be targeted in MS. But which part of the immune system? The immune system is very complex. There are various elements that are targeted by the various therapeutic interventions, clinically established or experimental. There is no such a thing as a magic bullet. There is not a specific tiny cellular type or a specific inflammatory cytokine that can be targeted that wipes out MS. And that's because there is a very high level of complexity in the immune system, in particular in the cytokine network, and that explains in part the redundancy and the inability to find this magic bullet. But in general, I think we need to be aware that we need to target the whole network, and we need to maintain a good balance in favor of anti-inflammatory factors. And that's the same thing that we should, in fact, aim with the microbiota, as I will show you later. And another very interesting discussion, talk that was held by Dr. Friedman not that long ago at CONI was, what is the reason for the failure of many MS trials? And we discussed some of the reasons. It's probably difficult to target a specific cytokines. Many of the anti-cytokine trials have failed. And that's because of the redundancy in the cytokine network and the lack of targeting the whole network. And also probably difficulties with predicting responder, non-responder status and selecting a specific intervention for the responders. But also first and foremost, I would say, and that is something I completely agree, is that many of these studies were based on EAE, the experimental model of MS, and mice are not humans, and that was the point that he made, and that is also very true. The big difference immunologically is that mice are very similar because they are inbred, whereas there is a lot of variability in humans that are outbred. But this is what actually eventually brought us to the microbiota. This is the eye opener, so to speak. Almost a decade ago, Professor Weckerle produced these mice that are transgenic and that develop spontaneous EAE, and he gave these mice to various laboratories, in particular in Germany, and the rate of spontaneous EAE in these mice was extremely variable. And these mice are genetically absolutely identical. And it turned out it was due to the microbiota. So that raised the awareness of microbiota. It was already, as you see here, it was a few years later, but it was already everywhere. Great interest in microbiota in health and disease, including multiple sclerosis. And so therefore it was obviously very worthwhile investigating it due to the well-known gut-brain interconnectivity, and the role of microbiota became a very important target for research. And we also know that the cells that are implicated in MS, the Th17 cells, develop in the gut, are instructed in the gut. But what is very important is that they are instructed by bacteria, so they develop with bacteria. This is even more fundamental because the gut microbiota also controls the whole microglia development. So the development of the normal microglia is under the control of the microbiota. Microglia being part of the brain. So the brain development is dependent of and function is dependent of microbiota. So that's very important. We all know, and that's been discussed and introduced, the human microbiome. What I want to emphasize here is that diet is its most important determinant in both humans and mice. This is a slide by Professor Yamamura from Japan, showing the dramatic increase in both MS here and Crohn's disease in Japan, and that's all due to the diet, and therefore the microbiota, as we discussed, it's the most important determinant of the microbiota is diet. And Professor Yamamura ended up showing very elegantly that there are species such as this Prevotella species. That is the species itself is not so important, but it is underrepresented in multiple sclerosis, and it happens to be one that is associated with regulation of the immune system. So it is relevant. This study, I won't quote all the studies, there are many. This study showed very convincingly the role of the microbiome in MS. This was a longitudinal, wonderful study by Dr. Howard Weiner's group that looked at microbiota in a context of also immunological MRI studies and clinical studies. They collected stool samples. It's not worth going into the great details. I'll show you some of the changes. So this is in MS, there's increase in this pro-inflammatory type of microbiota, such as this is Methanobacter, which produces methane and therefore can be measured in the breath. This is Akkermansia. As you see here, this is people with MS, and this is people with MS who are treated. So therefore, treatment reduces these pathogenic type of bacteria associated with pathogenesis of MS, and that suggests that there's a treatment effect. And that shows you the conclusion of this debate that microbiota is already a target for immunomodulatory therapy in MS. And this is an older study. There are some newer ones, but this is a review of all the studies looking at the role of microbiome. Yes, indeed, there are some species that keep coming up, including, as I showed you, the Akkermansia. But the species itself is not very important. It's just showing that there is a dysregulation in the balance between pro and anti-inflammatory microbiota elements is biased a little bit in MS in favor of the pro-inflammatory ones, and our aim is to reduce that and to restore the normal homeostatic balance. This is how one of 4 sheets of microbiome data looks like. And therefore, it's very complicated, it's very complex, and we should not think that, okay, we found that bacteria, we need to target that bacteria. No, but we need to target the whole, as I said, we need to restore the balance, we need to target the whole network. This is done naturally by some other infections, some more protective infections, such as helminth infections. That has been shown that helminth infections are associated with a more favorable microbiota, more an increased bacterial diversity, which is very important in the gut, and with a more regulatory type of anti-inflammatory microbiota. This is also associated, helminth infections, spontaneous natural infections with helminths in MS, are associated with a better course of MS and with increased regulatory T cells in MS. That drove us to do a trial in which we tried to treat MS with helminths. It was negative in the sense that the primary outcome measure was the number of new lesions on MRI. But if we had chosen, as people do these days in the days of NIDA, the proportion of people who have no new lesions, it would have been a positive study. As you see here, these are all the people who had 0 lesions in MS. That invalidated, that made the Mann-Whitney test that was pre-established much weaker because it had so many zeros. And that led to the decreased statistical power and to the negative results from that point of view. But it is remarkable that so many people compared to placebo had no new lesions. And that corresponded also with an increase in regulatory T cells, which was the second outcome measure, and it was positive. And as you see here, we checked the microbiota, and we showed that, as you see, the regulatory T cells go down in the placebo arm over 1 year, and also the microbial diversity in the gut in the placebo arm goes down, whereas it is maintained by infection. And as I mentioned, the bacterial diversity in the gut is associated with a healthy gut, and it's decreased in MS. So that is interesting because it shows this correlation. So we should not target specific taxa, but we should target this balance and tip it into in the favor of the anti-inflammatory responses. But DMTs that we use daily induce such changes in the microbiota. But what I would like to show you now, and so obviously the best way to target this whole network is to replace it. And there are studies, this is an older study in which there was some favorable responses in people receiving fecal transplantation for MS, in which some of the symptoms, some of the clinical aspects of MS improved. And this is a very nice N of 1, very well done study in MS with fecal transplantation with very clear microbiota studies showing again an increase, a steady increase in the bacterial diversity in the gut, and also an increase in this anti-inflammatory associated fecal bacterial process. The last half a minute, I would like to tell you the most important message today is that not only is the microbiota a target, but it determines the success of immunotherapies. This is work done over 10 years very elegantly by my former mentor, Professor Tim Carey, showing that immunotherapy for cancer depends on the microbiota. So the success of that depends on the microbiota. And here is a study last month in Nature Medicine showing that fecal microbiome, microbiota transplantation restores responsiveness to anti-PD-1 treatment in melanoma, and that the resistance to melanoma treatment is entirely dependent upon microbiota. So thank you for your attention. This is my message.
Dear colleagues, microbiome is really a hot topic in medicine and also in neuroscience. So I would like to thank the organizers for asking me to discuss this hot topic. And the key question of the day is: the microbiome is a therapeutic target in MS. I will defend the no option. So here my disclosures. So definitely the environment may play a major role in MS. It's clear we have huge amounts of data supporting that, probably related to the hygiene hypothesis. This hygiene hypothesis may be related to some key changes in the microbiota. So the microbiota is very complex. We know in neuroscience that we have a multi-directional microbiome gut-brain axis mediated by, importantly, the vagus nerve, bidirectional from brain to the gut and to the gut from to the brain, via acetylcholine as a key neuromediator. We have also a major role of the endocrine system, mainly via the cortisol axis. And of course, we have also a major role of the immune system. The gut is very rich concerning the immune system on the environment, and a close relationship with the mucosa, and so with the microbiota. Here, quite a complex slide, very busy slide, showing the complex interconnection between the nervous system and the microbiota and the immune system. So we have a downward control from the brain and the nervous system to the gut and to the immune system. Also, definitely, we have amounts of data showing that indeed the key role of the nervous system to induce some changes on the mucosa and to the immune system. At the opposite, the upward control from the immune system and the mucosa, and so possibly the microbiota, to induce some very strong changes to the functions of the nervous system. So some key change we also can discuss concerning the role of the microbiome in MS, induced by some key changes induced by antibiotics or probiotics. Also some key metabolites by the gut, or also some key experiments such as fecal microbiota transplantation of the data we have from the diet. We know some key changes induced by many mediators, to induce some key CNS changes concerning neuroinflammation. We are able to change the gut barrier permeability. Also indirectly, some key changes using probiotics, antibiotics, or metabolites from the gut concerning the integrity of the blood-brain barrier. Some key changes concerning the misbalance between the immune cells, pro versus anti-inflammatory profile, also concerning the humoral changes by IgA differentiation plasma cells of IgA binding of specific bacteria. So some key well-known experiments in EAE. We know that a broad spectrum of antibiotics are able to resolve mild EAE or attenuates severe EAE. Also concerning this experiment, one of the pioneers of these experiments, published by Berer et al. More than 10 years ago. You know, it's very difficult to have an EAE in germ-free mice. It's very easy to induce using different inductions an EAE in mice. What has been done here in these experiments, transfer of the microbiota from a mice with EAE to a naive mice, and it's able passively by transferring this microbiota from animal models to a germ-free naive mice. We have also the same EAE. Strong argument, of course, concerning the role of EAE. Also, interestingly, in also mice model, germ-free mice, to have the consequence of human fecal transplantation are also able to induce spontaneously an EAE in germ-free mice. From also, you see here in red the EAE incidence in mice, of course, by transplantation of human fecal from MS patients, and not possible or very few low incidence of EAE after transplantation from an healthy twin pair discordant for MS. So what is the composition of the gut microbiota and what are some of the key changes we have in MS? So you see the great categories of bacteria we have. Firmicutes, for example, the majority, with some key bacteria, you know, Clostridium or Lactobacillus, or maybe also a great category of bacteria is Bacteroides, for example, or Prevotella, also some Protobacteria or Actinobacteria. We know after different experiments that we have some bacteria are able to induce some key changes in the immune profile of T cells, pro-inflammatory profile, or on the opposite, some bacteria may have beneficial impacts, so beneficial bacteria into the gut to induce Treg cells, for example, or increase the secretion of IL-10. In MS, what we have, as you can see, definitely we have a gut dysbiosis in MS, that there is a consensus for that, a decrease in the diversity of the microbiome. Some experiments have shown a reduced Bacteroides profile with lower abundance of Prevotella or Bacteroides, with a higher abundance of certain Firmicutes such as Archemmansia or Doria. However, the long list of publications have shown that we have many discrepancies depending the timing of the disease, relapsing remitting, progressive MS, SPMS or PPMS, or even CIS, or even in pediatric MS, as shown by Tremlett and co-authors recently. So it's not clear if this dysbiosis involved neuroinflammation via the immune system or via some key changes concerning metabolites. For example, short-chain fatty acid, for example, we have some important difference concerning in MS with the composition of some fatty acids. Butyrate and precresol, for example, have increase in MS, can induce signal directly to OPC, but are respectively positive and negative regulators of OPC differentiation to oligodendrocytes. So complex changes, and we don't know the consequence concerning differentiation to oligodendrocytes, so maybe play some opposite role concerning remyelination. So if we look now at the preclinical studies testing antibiotics therapy, for example, in different EIE models, it's complex because we have different strains of mice, SGLG or Black Cyst, for example. We have different intervention and we have different results, sometimes in the good way, disease amelioration or disease worsening, or we have only with no clinical impact some changes of the at the immune levels or on some biomarkers, or sometimes only at specific level, at the spinal cord, for example, with very different interpretation concerning the mechanisms. And also now in MS, we have some pivotal trials, very few randomized controlled trial, usually short duration, small population, and open label study. So very preliminary data, sometimes in a good way, sometimes with no significant clinical changes. However, interestingly, we have some positive results concerning quality of life or patient-related outcomes concerning fatigue or depression, but very minor or no clear impact concerning relapse rate or EDSS changes, for example. Interestingly, also, if we look on some changes concerning the murine inflammation response, quite interesting, definitely in this key paper in PNAS a couple of years ago, some major changes involved by the changes on the microbiota, but however, very disappointing if we look at the remyelination, and in these experiments, no or minimal impact on remyelination or neuroprotection. Also interestingly, if we look now at some epidemiological data, we have no, to the best of my knowledge, no studies like that in MS. We have in Parkinson's disease. They show that indeed we have some key changes, not specific, but interesting changes concerning microbiota in Parkinson's disease. But we have partially the same changes in the healthy spouse of Parkinson's disease patients, and some common changes related to the environment, not only by the disease itself. So the key point is now not only to analyze the role of the disease, but also the role of the environment of the patients. So to conclude, I would like to say that definitely microbiota may modulate neuroinflammation. There is a consensus in different EIE models. Interesting findings from animal models, sometimes very far of MS patients. No consensus about microbiota signature in MS, even few common changes in the literature. No demonstration on remyelination or neuroprotections. We need to focus possibly more on the environment much more than the microbiota itself. It's not clear if some changes are causes or consequences. We have only few randomized controlled trials with very different products, no confirmatory studies with short duration. Many have biological outcomes or PRO, like well-being and emotional, no EDSS or changes related to disability. Studies with only single case with fecal microbiota transplantation and no clear impact on of DMTs. So, of course, it's a topic we need to improve our knowledge about microbiota on the role in neurodegenerative disease and neuroinflammatory disease like in MS. But however, it's not clear if definitely with the data we have today that microbiome is really a good target in MS. Thank you for your attention.
So thank you very much for both speakers who are now present for the discussion. I think one of the trick in the debate title was that it's a target, but is it also a useful target? I think this is perhaps the a key element. There have been questions from the audience, which is much appreciated. The first one was perhaps partially addressed by Patrick, but I could get both of your views on it. Are there any data about possible effect on disease progression when using probiotics alongside disease-modifying treatment? So the issue is probiotics. I think you had one table, Patrick, but I'm not sure what the effects tested were, whether that was solely on cell biology or also on clinical measures. So do you want to start, perhaps, Chris?
Chris, yes.
Hello. Yes, I mean, there are some data suggesting the first and quite convincing data comes from Professor Yamamura in Japan, who has shown that the microbiota is different between relapsing, remitting, and secondary progressive patients. And I think that's an important consideration when we're talking about perhaps targeting microbiota for disease-modifying treatment. And there are some data suggesting that drugs that are used for progressive MS have a different effect on the have an effect on the microbiota, and those specific changes, therefore, in the microbiome in the secondary progressive MS can be targeted. So possibly there is they can be therapeutically targeted.
Yeah.
The other thing that I had to say briefly, because I mentioned in my talk our Hooker trial, we had a small proportion of people with progressive MS in that trial, and we also find now at baseline, so before the treatment, that the composition of the microbiota at baseline, so before the trial started, was different between secondary progressive MS patients and relapsing remitting patients.
Right. Patrick, just on that point, anything to you want to add?
Yes, I think maybe possibly, as said Chris, however, to the best of my knowledge, and my feeling is that indeed it's possibly to have some improvement concerning some symptoms, constipations, some symptoms related to emotional controls, as demonstrated in others, neuropsychiatric disease, but my impression is no clear impact on the relapse rate or some key outcomes related to disability, time to walk, 9-hole peg test or EDSS. And also, if I've come back to the key point highlighted by Chris, for example, the use of parasites or hook-horn and so on, maybe possibly, but I'm not sure it's really related to the change in the microbiota itself, possibly related to the infection itself, chronic infection with parasite could also involve some key changes in the immune system, not possibly related to changes to the microbiota itself.
Very good, Patrick, here with adding a little sort of combat question right after your answer. Thank you. So I'm just going to go back to the point that I think you briefly made, both of you made, with respect to current disease-modifying treatments, their impact on the microbiota. And there was a question from Olaf Stuve regarding natalizumab. To what degree there's obviously a license for both MS as well as inflammatory bowel disease, whether some of the effects on MS are related to those affecting the microbiome. Is there good evidence for that?
Maybe I can answer first, if I can. I don't fully agree, because I think, first of all, I don't know if we have really a change in the composition of microbiome using natalizumab, first of all. And the consequence could be also related to the blockade of the immune system from the circulation to the mucosa. Because also I know the integrin is expressed on the mucosa, and also the blockade induced by natalizumab is probably related to the immune cells itself, and possibly nothing to do with the microbiome.
Okay, Chris?
Yeah, I mean, I agree with what Patrick said. It's there are some data, mostly from experimental systems, including the spontaneous EAE in primates, or an induced EAE in primates, that antibiotic modulate the disease, and at the same time change the bacterial composition of the microbiome, and also the virome, in the gut virome that is in the marmoset model, where there is some antibiotics or some other gut-targeted therapies lead to modulation and reduction of the EBV-like gamma herpes virus that has been involved in the pathogenesis of that particular model of EAE. But there isn't direct evidence from human data, as far as I know.
Yes, yeah, very good. I think what's obviously always a little bit sort of puzzling about the microbiome is that it's so broad and deep at the same time, right? So the question that we got through as well was, how specific can you be with targeting, as it were, the microbiome at the moment? It perhaps relates a little bit to what's your if you had a grant, right, unlimited grant, what trial would you undertake? What would you target? Perhaps Chris first.
So you're right. I mean, you cannot be very specific, and that's because with microbiota it's very difficult. It's a whole network. So you have to target the whole network. You cannot target a specific bacterium. It doesn't work like that. And so perhaps that's why the interventions have to be more holistic, such as fecal microbiota transfer, transplantation, or something that is a global, something that changes the population globally. And I don't think there's a specific
and possibly
relates bacterium that is responsible for the immunity.
Yes, and possibly also relates to a little bit to the previous debate we had about lifestyle. Patrick, any specific molecule you would be looking at?
Not really a specific molecule, but I think it could be possible to have a very personalized approach, to analyze the microbiome of one individual patients and try to see maybe as discussed by Chris, the profile, pro-inflammatory versus anti-inflammatory, the relative abundance of one category of Bacteroides versus Firmicutes, for example, and try to individually compensate, to induce a switch in the individual person. Of course, it could be very expensive. I don't know if it's realistic to do that or not, but maybe if unlimited grant, why not?
Very good. Excellent. So I just perhaps a final question to Chris. The hookworm study, you did find that this was, positive enough. Is there going to be a phase 3, do you think?
I'd like to say yes. I think that the data is sufficiently promising for a phase 3, but there are no specific plans. But we're thinking about it.
Okay.
Also, if there is one, I would change somehow the design of the study and the outcome measures.
Yeah.
I am a good player, Chris. I agree with you. I would like to be an investigator of this study. So
wonderful. Okay, so yes, I'm so sorry that we have to close the debate. I would at the end, terrific. Thank you very much for both speakers and the audience for engaging so well into this question. So the motion was the microbiome is a therapeutic target in MS. So what do you think about that now after you've heard the presentations and discussion? Your chance to vote is now. Interesting. So we had the original vote before the discussion was 58 to 42, and it is now 61 to 39. So congratulations, Chris, and
yes.
Well done, Patrick. But it's balanced enough for for both of you to go away into a nice and deserved weekend, no doubt. So thank you both.
Thank you.